Hello class, so welcome to this video. This video actually will cover all the questions of part 2 of the MCBS 4210 Immunology continuous assessment that we had last week. So total marks available for that part was overall on 10. So I will go through each question's marking criteria in details, and I will explain the reasoning behind the mark allocation. I will clarify what constitutes a full mark answer versus a partial credit answer. So don't come to me and complain if you have not watched the full video. And also I will highlight the most frequent errors seen in student scripts. So if you are a student watching this after receiving your mark script, use this video not only to understand where marks were lost, but also what the correct scientific answer look like. What an correct scientific answer look like. It will be a benefit for the exam. So this is not just about the marking, as you understood. It's not just about the marking. It's about deepening your understanding for immunology itself. So pay attention to details, how good answers look like, and do better during the exam. So let us begin with question 1. Question 1 actually was 2.5 marks, and it's broken into 4 marking criteria, each on 0.5 respectively, and each on 0.5 and 1 mark for the second part of the question. So we have we have 0.5, 0.5, 0.5, and then 1 marks for the triggers roles. Okay, so let's continue. The question says explain the concept of PAMs and DAMs and their role as trigger of innate immune recognition. So students must not only define what is PAMs and DAMs, but you should also explain what they do. That is how they trigger the innate immune recognition. And so that means if you only define the terms without addressing the whole has triggers part, you will lose actually the 1 marks allocated to that part. So this question integrates content from lecture on the innate immune system, and it also connect directly to the complement lecture, since complement activation through the alternative and lectin pathway is triggered by PAMs. Normally, if you are a good student, you should make the connection. Okay, you should make directly the connection. So let me go through each criterion. Okay, for criterion 1.1 on PAMs, after providing the full definition of these abbreviation PAMs, that is pathogen associated molecular pattern, the important word to bring actually when we talk about concept of PAMs is conserved. You can use conserved or invariant or conserved. I need to see that word. So this term is what explains actually why the innate immune system can recognize PAMs without a prior exposure. So because PAMs are structurally conserved across the entire class of microorganisms. So if a student writes PAMs correspond to molecules found on bacteria without the word conserved, that answer is incomplete or imprecise. So I will award a partial credit for that. But if the student provide examples, so this is just to complete the concept aspect. So the examples are helpful actually to award full credit, but not sufficient on their own without the definition. So the distinction between PAMs and antigen is a frequent source of confusion. Among the common error, students were mentioning PAMs has antigens produced by bacteria. Actually, this is not what is expected when you want to define that the concept of PAMs. PAMs are recognized by germline encoded PRR, that is um, pathogen uh, recognition receptors. These receptors are the same in every individual of the species, so they are inherited through the genome. But when we want, if, but antigen are opposite, or antigen are different. Antigen are recognized by the somatically rearranged receptor of adaptive immunity. So they are unique to each individual. So as we said in class, um, these pumps are shared by the whole species. That's a whole species, okay, when antigen are unique to each individual. So here you have the difference between the two, okay. And for the criterion 1 2, that is DAMS, after providing the definition, DAMS is damage associated molecular pattern. The required word to describe the concept of DAMS is endogenous. Yeah, they are endogenous molecules released by stressed, damaged, or necrotic host cells. So these are self molecule. They are not foreign, contrarily to pumps that will be reserved to microorganism that is non-self. Here is not foreign. So the clinical concept here is the sterile inflammation. Inflammation that will occur in the absence of any infection, and that inflammation will be triggered entirely by tissue damage releasing intracellular content. So the common error is observed is that students state that DAMs are foreign when it's not. Also, to confuse necrosis with apoptosis. Where is the difference? The necrosis versus apoptosis distinction is the really common error found in the student script. The difference we need to understand, because cells undergoing apoptosis, they do so in an orderly programmed fashion. So the cell membrane will remain intact until phagocytosis arrives. This is why apoptosis is described as immunologically silent or non-inflammatory, when necrosis will involve rapid membrane rupture and release of all intracellular content, including DAMs. So students who confuse these two forms of cell death in the concept of this question will not have a full marks on that question. In this second part of the question, so you were supposed to address the role of PAMs and DAMs as a trigger of innate immune recognition. So here criteria 1.3 and 1.4 will address the role of PAMs and DAMs as triggers of innate immune recognition. That means what happened after PAMs and DAMs are recognized. Once they are recognized, what happened? What are the different reactions that follow in the immune system? So the first thing required here is at least to name one of the pathogen recognition pattern, the PRR's families of the PR, at least one, for 0.5 marks. So you can see on the screen the 4 major family listed. You have toll-like receptors, node-like receptor, RIG-I-like receptors, and C-type lectin, as well as their location on the cells. So toll-like receptors are located on cell surface and on endosomes, and node-like receptors are cytosolic, as well RLRs, and CLRs receptors are actually cell surface. So students who will just write pathogen recognition receptor will recognize PAMs without naming any specific receptor. Families, that's also if you just call zero for this criterion, because asking you to name those is not extra, it's not just to disturb you. Actually, it's about being precise. As a biomedical science student, a future clinician, you need to know, for example, that toll-like receptor 4 will recognize LPS. You need to know all those details. So for criterion 1.4, actually that one is the most important one, since it carries the full 1 marks, and it is split into 2 equal half. The first 0.5 marks are for correctly describing the innate immune activation. So first you need to mention that during the innate, after recognition of PAMs by PRR, by dendritic cells, it will lead to their maturation, migration to lymph node, and activation of naive T cells. So this is the first part of the answer. In the second part of the answer, let's say the second part of the second part of the answer is where actually most of the students lose marks, because they describe only the innate response. They describe it well, but they forget to explain how it initiates adaptive immunity. Bringing the key concept of a second signal, that one was not was really rare among the script. For a T cells to be fully activated, it needs 2 signals. I think we explained it clearly in class. So you need signal 1 from the antigen MHC complex, and the signal 2 from co-stimulatory molecules like CD80 or CD86. So pathogen recognition receptor activation by PAMs will upregulate these co-stimulatory molecules on dendritic cells. And without this first PAM activation, without signal 2, T cells actually will encounter antigens. T cells that will encounter antigen become anergic rather than activated. So this is important safeguard against autoimmunity, and it is also directly relevant to why we need adjuvant in vaccines. Adjuvants actually they provide pump-like signal to make sure the signal 2 is delivered. As the signal 2 is really important for co-stimulatory molecules to intervene. So if the student have the whole explanation, not only limited himself to innate activation, but also bring the bridge to adaptive immunity, this is where you have the full 1 marks. It should include both innate activation and the bridge to adaptive immunity to have a complete answer on the role of PAMs as triggers of innate immune recognition. Okay, thank you. So let's move to the next question. The question 2 is worth 2.5 marks as well, and it is broken into 4 criteria of 0.5 marks each. So 3 criteria will correspond to 1 pathway each, and the 4th criteria will address the conversion effector function. As question 2 is outline the 3 complement activation pathway, their initiating triggers, and their conversion effector function. Here the word outline is in the question is an important directive. When you see outline, it does not require a deep explanation. It's not means explain. It's different from explain. Outline. So student don't need here to name every single protein in the cascade, but they do need to identify the trigger first, the keys of recognition molecules, and the C3 converter generated. So outline that are too superficial. For example, classical power is activated by antibodies with nothing further. You will have less than the dedicated marks. It's not just about mentioning like generally antibodies will activate classical pathway, but which antibody precisely, which molecule exactly. And the word convergent in this question, convergent, the convergent effector function. So the word convergent in the question is also a marker for students if they use that word in their answer. They have understood that all three pathways lead to the same downstream effectors mechanism. A student will describe three pathways but then list three separate set of effector function has not understood convergence. Convergence means all three coming to one terminal end. So that means you will lose the marks dedicated for that if at the end you don't have a convergent effector function of the three pathway. Okay, let's go directly to the correction of this question too. Okay, here on the slide you can see the three activation pathway and well detailed and defined in criteria 2.1, 2.2 and 2.3. So I will go through each of the three pathway in turn, pointing to the required terms and the most common error. So in red you have the most common error. Pay attention to it. So for the criterion 2.1, the classical pathway, that is on 0.5 mark. Note that to have the 0.5 marks, you need not only to mention the required terms, but also to be able to outline the element, the key element. So in the classical pathway, the two required terms that should appear in your script for you to have the 0.5 at least is IgM or IgG has the triggers of that classical pathway. But not only you need IgM or IgG, but also C1q. They are all the recognition molecules. So C1q is recognition molecule. So students who write antibodies activate complement without specifying the immunoglobulin class and without naming C1q score a maximum of 0.25. That means half of the mark dedicated for that question. The reason C1q is required is that it is the molecular mechanism. It's the base molecular mechanism. So you have C1q is what physically bridge with the antibody antigen complex. So you need antibody antigen complex formation and to bridge to bind with C1q to have the complement activation. So the C3 converters generated, that will be C4B2A, is worthy if mentioned, though not strictly required for the outline. In outline here you need antibody antigen complex formation and you need the C1q that will be the bridge with the complement pathway activation, that particular complement pathway activation. So one nuance I want to mark positively, if a student mentioned, for example, that IgM is more efficient than IgG because of its pentameric structure, providing multiple simultaneously FC region for C1q binding, that demonstrates a really good understanding of the principle, the mechanism here, and should be there therefore awarded full marks for this criterion. Even if other details are missing, but if this precision is formalized, yeah, applause for your student. Okay, criterion 2.2, the lectin pathway. Here the most important term is MBL. Either you should indicate the triggers are MBL for mannose binding lectin. You can also use mannose or ficolin as the recognition molecules. But you also have MASP1 or MSSP2 has the activating protease, this enzyme, because effectively MBL will activate the protease MASP1 and MSSP2 that are analogous to C1R or C1S from the classical pathway. So same, it will lead to the C3 convertase into C4-2A as in the classical pathway. So this one is antibody independent, but it's a true first line innate defense. This one is a true first line innate defense. So bacteria they will expose the mannose arrays because the lack of the Golgi processing machinery that all mammalian cells use to add sialic acid at the terminal position of glycan chain. So this is what makes a bacterial surface recognizable as foreign by MBL without requiring any prior adaptive immunity response. Okay, this is for now the criterion 2.3, the alternative pathway. Yeah, this is the most conceptually complex and the one that students most frequently get wrong. Yeah, if you remember in class we mentioned about the triggers that can be it's a bit confusing. We mentioned chemical reaction. Chemical reaction can be triggers, but in books we have C3 ticks over or spontaneous hydrolysis as the mechanism. The word that I expected here to see is factor B and factor D has the activating proteins, and popagine has a stabilizer of C3b3b. C3b yeah, maybe I should have added the design or a sketch of these three pathways so that we have but you can go back to your notes. Actually, you have it already in class in the lecture notes, so you can, when I'm talking, you can use the figure of this three pathway to see very exactly what I'm talking about. So the most important conceptual error to flag here in this particular pathway is that this pathway has no specific triggers. So it's run continuously, it is always on at low level. What determines whether it leads to a full complement activation is not activation, it's not really a trigger. It is whether regular proteins are present to shut it down. So it's always on. So on self cells, factor H or DAF will rapidly inactivate C3b, but on pathogen which lack these regulatory proteins, C3b survive, will recruit factor B and form the C3bBB convertase. That is a fundamentally different logic from the classical and lectin pathway. So students would describe the alternative pathway as activated by LPS or by bacterial molecules or bacterial surface, are describing the outcome, not the mechanism, not the process. They are describing the outcome, because bacterial surface do support alternative pathway amplification, but they are missing the student are describing this are missing the mechanism inside that pathway um, activation. Okay, so and also that pathway, the discrimination between self and non-self is regulatory here. It's trigger based. It's regulatory, not trigger based. So if we need to keep something, the take home message from this pathway is it's always on. So there is nothing that is really the triggers of that pathway. But let's say this element that we can cite, the C3 tick over, the spontaneous hydrolysis or properdin stabilization, they will amplify the loop. Okay. They will not start it, they will amplify it. So this is all you need for this question 2. Let's move to question 3. Oh, I talk a lot on that first part. I almost forget the the second part of the question, actually, that was about convergent effector functions. So let's close it first before moving to question 3. That is criterion 2.4. That is about convergent effector function. So what all the three pathway produce when C3 is cleaved. This is 0.5 marks if you have full for all the three functions correctly identified. So let me explain why I require all the three for full marks. The question used the word effector function, so in the plural, and the word convergent signal, that the student should describe what all pathways share in common. So the three effector functions are opsonization, inflammation, and lysis through the MAC formation. So that one need to stand on your paper. They are taught as a triad in every immunology textbook. You can find it, them three as the terminal ending. So a student will remember only, for example, only the MAC has remembered the terminal, but numerically the least important of the three. In quantitative term, far more complement activity result in opsonization than in MAC assembly. So to note, for your knowledge, MAC lysis gram-negative bacteria easier than gram-positive bacteria, as the latter are resistant because their thick peptidoglycan wall prevents MAC insertion into the lipid bilayer. When for regarding inflammation, so you have this small description in each box that you can read on inflammation or anaphylatoxin. Student mentioned histamine release without student that mentioned histamine release that without explaining the mechanism is not complete. So the mechanism is C3A and C5A bind G proteins coupled receptors on mast cells and basophils, triggering degranulation and histamine release. I want the receptor binding step to be mentioned, even briefly, for you to have the full credit on this question. And regarding opsonization, how to describe it. So students should ideally mention CR1, that is complement receptor 1, as a phagocyte receptor that binds the C3b. So if they write only phagocyte engulf C3b coated bacteria without naming which receptor is responsible for that coating, I will accept the partial credit, but the answer is technically incomplete. So for your scientific correctness, just bring always that details, name the molecules or receptors involved. And the thousand-fold enhancement of phagocytosis figure is a clinically meaningful number that will demonstrate the understanding of the quantitative importance of opsonization. So here you have all the three pathway will converge to a C3 cleavage, and this convergence will produce three effector output. This is how you should write your answer. And now list them, list the effector output. It can be opsonization, inflammation, or lysis using the MAC formation. And for each brief definition of what is opsonization, who are involved: C3b deposits covalently on pathogen surface, or in inflammation we have C3a and C3-5 that are anaphylatoxin, and in lysis we have C5b nucleate that will lead to connecting the C6 to C7 to C8 and then the C9s, all the C9s, to form the MAC, that the attacking complex membrane. Okay, so now we can go to question 3. Okay, question 3. Name the major mediators of acute inflammation and state their principal effect. So this question 3 is the most like marked rich question with full 5 marks. So the marking rules here is simple and absolutely consistent with previous ones. So if you name a mediator correctly, you give at least one accurate principal effect, then you earn 0.5 times 2. So the critical rules for the 0 marks: if you see 0, that means you give vague effect, score nothing. That if you just give a vague effect, that that it will score nothing. And a student will write, for example, histamine cause inflammation. You are right for the mediators, but for the effect it's just vague. So you will earn here 0.5 marks. So a student will write instead histamine, vasodilatation, and increased vascular permeability. That is correct, so you have your 1 marks. So the distinction between cause inflammation and saying vasodilatation and increasing of vascular permeability, this is exactly the distinction between knowing a word and understanding a mechanism. So make sure you understand the mechanism for the exam, because for the CA now it's late. So the expected performance by student level, the expected performance I will expect a well-prepared year 3 biomedical science student to correctly be able to list 7 to 9 mediators effect pairs. So in this case, the scoring will be between 3.5 to 4.5. I don't actually ask you to list 7 here. 5 is okay, but I mean you are expected to be able to list 7 at least. So an excellent student should achieve all the 10 on 10, but you are being asked 5. An average student will correctly identify 4 to 5 pair. So the continuous assessment was set for average student, and then normally you should earn 2 to 2.5, but in this case you have 5.5, 5 on 5, and which I consider as an acceptable, a good performance for this question. So a student who will score 2 has either not studied the topics or has listed names without effect. So if you are able to list the 10 mediators and now being confused on the effect, go back, read your notes really carefully as the exam are coming soon. Prepare yourself very well and do better the next time. Okay. Okay, so here I listed mediators from this first slide. You will see mediator 1 to 7, but there are more mediators. So you should be able to know each of them with their effect and their localization. Not only for the CA, but for your knowledge to be a good clinics. And the first one that actually appear also the most is histamine. This is the most commonly named mediators, and most students will correctly identify vasodilatation and increase vascular permeability with this one. So not a big deal. So you have serotonin coming after that for vasoconstriction, local vasodilatation, increased permeability. These are the effects induced by this mediator particularly. So you have the table here. Please take the time, because here you have a really concise and a table format. Maybe this one will be easier for you to read compared to the ones the you have in your lecture note that was full paragraph description of mediators and their effect. So here you have a synthesized version. Please take the time to go through to read each of them, because it's really long. I don't want to stand and just read them for you. So please take you can reproduce this table so that you have a really concise form to read, to prepare with for the exam. And so just know that for you to have 5 marks, you need to have a combo of at least 5 of the mediator with the right effect. If you have a correct answer with the mediator, then it's 0.5. If the effect is not okay, it's 0 on the effect. So if you have a pairs of mediator and effects, right, this is 1 marks. You need to have 5 to have the full 5 marks on that question. So here is the second part of the table. You have more mediators and their effect. So please take the time, read it, make sure you know them all with their respective effect, and exert yourself, try it, do it on your own. Check your memory, see if you can effectively be able to list at least 7 with the correct effect, and you will be the perfect immunologist. Okay.